Right, so now we know about the central nervous system, and we know that it consists of our brain stem, and our spinal cord. But what about the rest of the nervous system? Well, that's what I'm here to show you. And so the peripheral nervous system really sort of starts with the spinal nerve. And before we can talk any more about the peripheral nervous system, we need to talk about how a spinal nerve's actually formed. And so you can see that here we've got several different nerves coming out of the spinal cord that you can see here. And I'll just quickly orientate you so you know where we're talking about as we go. Now this is the anterior surface of the spinal cord, and this is the posterior surface of the spinal cord. Here you can see the anterior median fissure. Difficult to see the posterior median sulcus there, but you can see it's very thin, extending to the surface. Here you can see the ventral horn of the grey matter, and it's sort of short and bulbous. And here you can see the dorsal horn of the grey matter, and it's long and slender. So hopefully that gives you an anterior and posterior orientation to work with. Now what you can see here is named the ventral rootlets. And if you remember back to the previous lecture, that the cell bodies of the motor neurons, those large multipolar neurons, were located here in the ventral horn. So the axons from these motor neurons are going to pass through these ventral rootlets. And because these axons are seen as carrying information away from the central nervous system, we say that they are efferent axons, that are efferent, okay? You'll hear this term later. Now, that means that the ventral rootlets are entirely motor because they are axons from the motor neurons, so they're exclusively motor. Then these rootlets coalesce, and they're going to coalesce into this structure, and we're going to call that the ventral root or the anterior root. Again, all motor. Now on the opposite side, we have the dorsal rootlets. And the dorsal rootlets is going to bring sensory information back to this. And this is the dorsal horn of the grey matter. So the dorsal rootlets are exclusively sensory. And when we say something is coming back toward the central nervous system, we call it an afferent neuron. So the motor is efferent, and the sensory information is going to be afferent. Now these dorsal rootlets have come from this structure, this nerve, and we're going to call that the dorsal root. Again, exclusively sensory. Now the cell bodies for these axons that you can, there we go, there's my mouse back. Now, the cell bodies for the axons that are contained within the dorsal rootlets and the dorsal root are located here. And this is a little swelling that you'll see just lateral of the dorsal root, and we call that the dorsal root ganglion. And now remember that a collection of cell bodies in the peripheral nervous system was known as a ganglion. And these are going to be those pseudo-unipolar neurons. And so they would have collected information somewhere out around the skin, and the action potential would have taken it back along here, and it would have bypassed that cell body, gone back through the dorsal rootlets, and into the dorsal horn. Now you can see here that the ventral root and the dorsal root actually join into this short little segment, and that segment's usually only about 1 centimetre long, and this segment is called the spinal nerve, and we have 31 pairs of spinal nerves. As I said, very short, and they almost immediately branch off. So from the spinal nerve, we have a dorsal branch, and we're going to call that the dorsal primary ramus, sometimes shortened to dorsal ramus, and then we're going to have a ventral branch, or an anterior branch. And this is called the ventral primary ramus, or the anterior primary ramus. Again, ventral and anterior are synonymous, dorsal and posterior are synonymous. Now the key thing to remember with the dorsal and ventral primary rami is the plural for ramus, is that they are mixed motor and sensory. So it's at this point medial to the spinal nerve that they split into sensory and motor, but lateral to that, in the spinal nerve and in the rami, they are mixed sensory and motor. So just to summarize, the ventral rootlets, which are here, and the ventral root is motor, which is classified as being efferent because its information is going away from the central nervous system. The dorsal rootlets, which are here, and the dorsal root, which is here, is afferent because it contains sensory information, and the sensory information is going back towards the central nervous system. Now the nerves that are mixed here are going to be the spinal nerve, which is here, the dorsal primary ramus, which is there, and the ventral primary ramus, which is there. Now we need to look at where they actually go. So if we look in this diagram, I'm just going to show you again how the arrangement goes, make you familiar with some things that you may have forgotten from the last lecture, and show you where they actually go. So here's the dural sac that you can see here. Again, the outer tough layer of the meninges, and you can see some of the arachnoid in there. The pia will be in contact with the spinal cord. You can see here we have an epidural space. Here's the anterior epidural space. Here's the posterior epidural space, and they are filled with epidural fat. So that means that we can see the spinal cord in the center here, and we can see the anterior median fissure here, and the posterior median sulcus just there. Posterior lateral sulcus is a little bit hard to see on this image. Here's the ventral horn, here's the dorsal horn. From the ventral horn, we're going to have the axons going out into the ventral rootlets, which are here, into the ventral root, into the common spinal nerve that you can see here. Okay. From the dorsal horn, we're going to have the dorsal rootlets coming back into that dorsal horn, and we're going to have the dorsal root, which is here. And the cell bodies for these sensory neurons of the dorsal root are going to be here in the dorsal root ganglion, that little swelling that you can see there. Okay. Now you can see that they come into a common spinal nerve. This is mixed motor and sensory. And we can see that we have a dorsal primary ramus here, and we can see it more clearly here. In fact, here we have the dorsal primary ramus, and you can see that this is heading posteriorly. So the dorsal primary ramus is going to supply the skin of the back and the muscles that move the spine, which are referred to as the postvertebral muscles. And here we can see the ventral primary ramus, the ventral primary ramus. Where's that going to go? Well, everywhere else. And we're going to be looking later in the lecture on what the fate is of the ventral primary ramus. But first, I'd like to talk a little bit about the different spinal nerves. So far we've just looked at one spinal nerve, but there are in fact 31 pairs. So let's look at them. So we have 8 cervical spinal nerves, we have 12 thoracic spinal nerves, we have 5 lumbar spinal nerves, we have 5 sacral spinal nerves, and we have 1 coccygeal nerve. Now, what's important to see here is that the nerves don't always equal the number of vertebrae that we have. So we have 7 cervical vertebrae, but we have 8 cervical nerves. And how does this work? Well, what happens is that in the cervical spine, the nerves exit superior to their named vertebra. So here we have C1, and here we have the C1 nerve, spinal nerve. C2, C2 spinal nerve. C3, C3 spinal nerve. And so on until we get to C7. And C7 has 2 spinal nerves associated with it. Superior to the C7 vertebra, we have the C7 spinal nerve, and inferior to C7, we have the C8 spinal nerve. Now, inferior to the C8 spinal nerve, all of the spinal nerves exit inferior to their named vertebra. So we have T1 vertebra here, and then we have T1 spinal nerve coming inferior. And likewise, all the way down for the remainder of the spinal canal, we have that arrangement. And we're going to be talking again about the fate of those different spinal nerves. But we need to first have a bit of a look at the cross-section of the spinal nerve in a microscopic power. So let's review this. So we can see that here we have our axon, and this is obviously a myelinated axon because we have a Schwann cell and we have a myelin sheath here. So this is the nucleus of the Schwann cell. And then surrounding this myelin sheath and axon is this connective tissue called the endoneurium, and endo means inside. And so you can see there's lots of endoneuria surrounding a lot of axons that you can see here. And then the axons are all grouped together like electrical wire. And when they're grouped together, they're called a fascicle. And then there's another connective tissue that will wrap around the fascicle, sort of keeping all these electrical wires, the axons, together. And that's called the perineurium. And the peri, part of perineurium, the peri prefix, means around. So it's going to wrap around the fascicle. Then, finally wrapping around the whole spinal nerve is this, and this is the epineurium. And epi, as you might remember, means above or upon. And so this is going to be the connective tissue layer upon everything else. Okay, so we've just cut transversely through the spinal nerve there, and we can see how the spinal nerve is actually packaged. Now from that spinal nerve, I said we had a ventral primary ramus and a dorsal primary ramus. And I want to talk to you a little bit about the fate of the ventral primary ramus. And there's two ways they can go. One is they can remain segmented and not join with anything else and just go to different parts of the body, or they can be integrated. And when they're integrated, they form what's called a plexus. And so this is where the axons of these ventral rami become all sort of mixed up. And so axons, say from section A of the spinal cord, this is just an arbitrary naming system, section A of the spinal cord, might end up in 1, 2, 3 different areas of the body. And likewise, from section B, you might getting axons from section B of the spinal cord, ending up at 1, 2, 3, 4 areas of the body. So this can get quite confusing when we actually look at the different formation of the plexuses and what spinal nerves contribute to what area of the plexuses. But we'll talk about that later in the semester. So let's talk about the fate of specific ventral rami and whether they contribute to a plexus or not. So the C1 to 4 ventral rami are going to become part of this cervical plexus. And you can see the cervical plexus just here. And we're not going to be looking at the cervical plexus much throughout the semester. It's a bit out of the scope of this unit. The C5 to T1 ventral rami are going to be able to form the brachial plexus. And the brachial plexus will be the structure that supplies the upper limb. The T2 to T12 ventral rami are segmented intercostal nerves. So they're not actually going to form a plexus whatsoever. They're just going to continue in their intercostal direction. Costal means rib, inter means between, so they're going to be between the ribs. L1 to S4 is going to become the lumbosacral plexus. And something that I haven't mentioned on this slide, but just for the sake of completeness, the coccygeal nerve is by itself and it doesn't form a plexus. So we've just been talking about the fate of the ventral rami here, but we also need to talk about its counterpart, the dorsal primary ramus. Now, the dorsal primary ramus is always segmented. And what it will do is, as we said, it will go on to supply the muscles of the back and the skin of the back. So having said all this, we need to start thinking about how we're going to view these nerves in the lab and what they actually look like. And this is what they look like. Now remember, because the nomenclature can get quite confusing, Neurons are microscopic and nerves are macroscopic. So nerves you can see with the naked eye, neurons you can't. You can only see with a microscope. So here's the sciatic nerve, and it's the largest nerve in the body. And you'll see that the way to differentiate it from something like a tendon, for example, is that a tendon will be thick and it will be taut and it will be rigid and it will of course be attached to a muscle. But a nerve will not be that way at all. A nerve does not have much tensile strength, so it'll actually be quite floppy, and it will be soft to the touch, and it will be quite distensible. So that's the way that you can identify a nerve from a tendon on a body. So I encourage you, when we go into the wet labs this week, to actually pick up a nerve, pick up a tendon, and try and identify the textural differences, because this is part of anatomy, is the palpatory textural feel of the different tissues. I imagine you'll probably have some questions from the peripheral nervous system lecture that we've just gone through, and that's totally fine. So please write them down and bring them to me in lab, and I'm sure everyone else in the class has the same questions, so we can work through them together. Thank you.